Swayam Prabha. Digital India. Educated India.
Okay, so this was about enzyme kinetics and enzyme inhibition kinetics. In the next 45 minutes, I will give you an introduction to a field of chemistry that that has been there for a long time, but it is a very interdisciplinary in nature. It is called so this is a contemporary field. So did any one of you have any course or understand or know anything about this field called biophysical chemistry? You never had any course on this? Okay. So for some reason in some of the universities it is not taught, but in a lot of other universities this is actually a proper course, or like just like your electrochemistry, just like your spectroscopy and other things. So biophysical chemistry is a proper course. Okay. So it is an interdisciplinary science. So what in the next 40 minutes, or 45 minutes, what I'll do is, what is this field about? What are the problems that this field studies? What are the techniques that we use? Okay, just to give you introduction to this field. I'm not going to give you any particular problem, or I'm not going to tell you about any specific example, but I'll only give you an overview of what this field is, what are the problems and what are the techniques, and where is the research in this field going. Okay, so as you can see, biophysical chemistry. So as you can see here, so what biophysical chemistry talks to you about, I am not going to talk about single molecule biophysics, but what biophysical talks to you about, you are trying to understand the chemical properties, mostly physical chemical the physical chemistry properties or physicochemical properties using advanced instrumentation of biomolecules. Okay, so you basically try to understand, so do you know what are biomolecules, right? So we are trying to understand, or what people try to understand is, if you take a field of physical chemistry, right? So what are the properties that generally people study in physical chemistry? You calculate the thermodynamic properties like delta G, delta H, delta S. So you basically try to understand the energetics. Thermodynamics, you try to understand the energetics of interactions, right? So you try to understand the energetics of interactions, and what else you try to understand? Quantum. What will quantum give you? So till now we have done something else. What else we have done other than thermodynamics and energetics? We determine kinetics, right? So you determine the rates of the reaction, you determine the basically kinetics of the reaction. And what else do you have in the physical chemistry? You determine the structures of the molecules, right? Using spectroscopic methods. You can use optical or magnetic or other kind of methods. You can determine the structures of the molecules, right? So you can determine the thermodynamics of the molecules. You can determine thermodynamics and energetics, okay, and kinetics of the molecules, then structure of the molecules, and what else we can do? We can also determine what is called dynamics of the molecules. Do you know what is dynamics? What is dynamics? You have heard of molecular dynamics? Okay, what is dynamics? Movement. So, movement of the bonds, right? So basically when you're talking about molecular dynamics, you're talking about the movement of the, in this case we are not talking about the overall movement of the molecules, but movement of the bonds, right? You will have all these vibrational modes and other kind of modes, but still you're talking about the movement of the molecules. So biophysical chemistry basically deals with determining the energetics, kinetics, structure and dynamics of biological molecules. Okay, this is what biophysical chemistry is about. And what are biological molecules? Proteins, enzymes, carbohydrates, glucose. Glucose again carbohydrate, no? Molecular interactions, yes, you can always study molecular interactions also. you can understand how they are interacting, right? So you can also study molecular interactions. Again, interactions means what? Energetics only. No? When something is interacting, you are monitoring the energy. So you monitor the energetics there, right? So proteins, enzymes, or proteins and enzymes are similar. Then you have nucleic acids, right? Nucleic acids, carbohydrates, lipids, right? So these are all classified as biomolecules. So you all know organic chemistry, right? They're all dealing with small molecules, and biomolecules means that are found inside biological systems, that are excessively found. All the molecules are found somewhere or the other, but your entire biological life or biological system is mostly made up of these molecules: DNA or nucleic acids, proteins, carbohydrates, lipids, right? So mostly it is made of this. So these are called biological molecules. So when you study the physical properties that I mentioned just now, right? All the four or five properties that I mentioned: energetics, structure, kinetics, what else did I say? No, thermodynamics is energetics only. Dynamics of these biomolecules, that is nothing but your biophysical chemistry. Okay, so this is a field that has been there for a long time, but people have for some reasons you don't really classify it as biophysical thing, but it's still a biophysical chemistry, because you are looking, instead of looking at smaller molecules, you are looking at a much larger molecules. So biological molecules have a lot of heterogeneity and they are much larger. Okay, so it is something like this. It's a bridge between. So you actually look at how the how and why the physical processes of biomolecules happen. Okay, this is also in some places called molecular biophysics. So if you look at, you all are you aware of IISc, Indian Institute of Science? So there is a special department called molecular biophysics. It is nothing but biophysical chemistry, right? Biophysical chemistry is nothing but biophysical properties of small molecules at a molecular level. So it is nothing but, in the other way it is called molecular biophysics. That means you are trying to understand the physical properties of biological molecules at a molecular level. Okay, you are not looking at a cell, or you are not looking at an organism, or you are not looking at a much bigger thing. You are only looking at a molecular level. So that is called molecular biophysics. It is also called as biophysical chemistry. Okay, let me quickly show you. You are all aware of these biomolecules, right? DNA, nucleic acids, and then proteins and enzymes and other things. I'm not going to tell you about this, but I am only going to tell you about the problems that this field actually looks at. So I have kind of made a cheat sheet kind of thing. As I told you, so this is you can generate by ChatGPT or the regular features of this. So the major focus areas is basically you try to determine the structures of proteins, which is, or biomolecules. Does anybody know how do you determine the structure of molecules? NMR, okay. IR will give you functional groups, okay. One more common thing, X-ray diffraction, XRD, right? So XRD, and because these are slightly larger molecules, you can also use electron microscope. So you can so I have shown you large examples, right? Like I have drawn you cartoons of enzymes, but they all have proper enzyme structures, right? For example, these are all structures of proteins, right? These are all structures of proteins. So how do you determine these structures of proteins? So the protein structures are determined. So you basically determine the structure of biomolecules using NMR, not UV visible fluorescence, but X-ray crystallography and cryo-electron microscope. Okay, the most recent advanced technique is cryo-electron microscope. So you are basically determining the structures of the molecules. That means how they look. So for you to design any drug, or for you to understand the mechanism of action, you need to know how the molecule looks like, right? It is not a simple organic molecule where you can determine the NMR structure and NMR spectra and determine it. You need to do a multi-dimensional NMR, not a single dimensional NMR. You know there is something called 2D NMR, 3D NMR, multi-dimensional NMR. So you have to do multi-dimensional NMR to determine the structure of proteins or DNA. And you can also use X-ray crystallography, which is based on your X-ray diffraction, and then a cryo-EM, which is electron microscope, okay, because these are large molecules. So how big do you think these biomolecules are? Suppose I have a protein. What do you think is the typical size of a protein? Daltons is the molecular weight. I'm talking about size. Nanometer, meter, centimeter, kilometer. What is the size of a simple small molecules like suppose you have a hydrogen or a carbon dioxide. What do you think is this regular size? Or you have a CC bond, right? A CC bond is how much? Suppose C single bond C, okay. What is the size of a single CC bond? Or if you just have CC, right? What is the bond length? Angstrom. How many angstroms? 1.09. It's around 1.56 angstroms, no? Okay, so you're talking about angstroms for a single bond. Now biomolecules are slightly larger, right? So biomolecules range anywhere between few nanometers to tens of nanometers. So you can use a high resolution electron microscope to determine that. So you can use basically X-ray crystallography, cryo-EM and multi-dimensional NMR to determine the structure of biomolecules, okay. Now this is structure. The other thing I said is energetics, right? Thermodynamics. So you can calculate the thermodynamics and kinetics of this. How do you use that? So you can use instruments like ITC, isothermal calorimetry. So have you guys gone around the department to see various instruments? Did you look at ITC and DSC? So isothermal calorimetry, what does it give you? Isothermal calorimetry means it will give you the basically the enthalpy and the entropy of a reaction. Mostly it will determine the enthalpy. So it will give you the energetics of the reaction. So it can give you DSC and ITC. So you can determine the energetics of the reaction using these techniques ITC and DSC, okay. Then you can determine the kinetics of a reaction using techniques like stop flow method and relaxation method. So you can understand how much like I said the energetics of a reaction or kinetics of a reaction, large molecules using various methods. And you can determine the dynamics. Dynamics means, as I said, there are dynamics is where there is a motion, molecular motion, right? So we just showed these things, right? I told that you have a polypeptide that folds into a tertiary structure. You have a polypeptide that takes a secondary structure that folds into a tertiary structure and forms a protein. So there is a motion involved in this, right? In this entire folding process, there is this entire process of protein folding. That means there is a change in the dynamics of this entire molecule. So that is called dynamics, where you are looking at the motion of the molecules. And as we also saw in the enzyme kinetics, the enzymes, you remember what the first day, yesterday I said there are two mechanisms for enzyme binding. What are they? Lock and key model and induced fit model. Lock and key model means you have a fixed enzyme, the substrate comes and fit. Whereas induced means basically your substrate comes, but then the enzyme kind of changes its shape so that it tightly binds. So that means there is a dynamics involved because the enzyme is changing its shape to fit to the substrate. That means there is a motion of the enzyme, right? So that motion of the enzyme is nothing but dynamics. So you can study protein dynamics. So both protein folding as well as in enzyme substrate, you can study the dynamics of this protein. And to study the dynamics, you generally use techniques like fluorescence, UV visible spectroscopy, and you can also do molecular dynamic simulations, okay, which is basically computational method, molecular dynamic simulations, or mostly you use fluorescence, UV visible spectroscopy, because these two cannot give you dynamics. These are static pictures, right? Cryo-EM can in a part give, but crystallography means you are crystallizing it, right? And then taking the structure. So you will only get the structure, you will not get the dynamics. NMR can give you dynamics. NMR, UV visible, fluorescence and molecular dynamics, these all can give you information on dynamics, that is how the protein is folding or how the enzyme is kind of inducing or fitting it to the substrate. Those things can be studied. And then you can study what is called molecular interactions, right? So as you mentioned, basically you are looking at the interactions between the different molecules. So there is something called, I'll tell you different problems that are there in this. So there is a problem of aggregation, there is a problem of binding, and these things can be studied by any of these methods, either ITC or fluorescence and these things. So these things can actually give you the information on the molecular interactions, okay. So now there are various problems that are studied in biophysical chemistry. First thing is structure function relationships. So you know about proteins. So do you know that there is a term called proteins are called as workhorses of the cell. Do you know that? That means anything that is going on in your body, right? For example, a muscle has to contract or expand, or if there is a transmission of chemical molecules, for example, your oxygen has to be carried from one part to other, right? If there are myoglobin, hemoglobin, there are titin proteins. Even if I am talking, basically there are proteins in your ears that are actually, so basically there are vibrations that are going on, right? This is sound waves, right? The sound waves will touch, there are proteins, and that results in the vibration of the proteins, and depending on the vibration of the proteins, your information is going to your brain, right? So basically there are proteins in the ear, there are mechanosensitive proteins in the cell. So you take any action in the body or in the cell, all these are done by proteins, right? DNA is, it is like your DNA is like a code. DNA will code to proteins, but all the functions of a body are done by proteins. Any function, either it be mechanical functions or most electrical functions or kind of acoustic functions or most of the functions. For example, I think I have something like this. So you have a lot of proteins that are hormones, that are enzymes that give you structural identity. Like for example, titin. So you have a protein called titin in your body, which is a it's a muscle protein. Okay, if you do you know, like your heart expands and contracts and expands and contracts. Did anybody? So if you have a spring, right? If you keep on doing it, at some point it will lose its elasticity, right? But there are muscles, inside the muscles there are proteins, right, which are called which belongs to the titin family, which kind of expand, contract. There are springs like proteins that kind of, these are all alpha helical proteins, expand, contract, expand, contract. So they will give you the support, right? So your structural support. And then there are ion channels. So you know that there are like a lot of, in the cell there are a lot of ions that are actually transported. There is sodium ion pump, there is a potassium ion. You know that if you take if you get dehydrated, right, you fall sick. Why do you get dehydrated when you fall sick? Because the electrolytes in your body go down. That means then what you do, you take something called electrol powder, right, which has a lot of ions, sodium and potassium. So there are actually these proteins that allow these ions to reach to their correct place. These are called ion channel pumps. So proteins act as ion channel pumps. Okay, proteins act as structures, proteins act as enzymes, proteins act as hormones. For example, your insulin, right? It's a hormone. So it acts as proteins. So they're derived, there are smaller peptides, but so proteins are also acting as antibodies, right? For example, a simple, if you are taking if you're having, you all took vaccines, I guess, right? Yeah, how do vaccines work? Most of the vaccines, they produce what are called antibodies, right? So basically they produce antibodies and they try to kind of, when a new virus or new viral entry happens, you're already giving it pre-information so that there are antibodies that can fight against it, right? So most of the antibodies are also proteins. So there are millions of proteins, right? So to understand the structure of these proteins, every protein has a different structure. To understand, if you want to design a drug, you need to know the structure of the protein, right? To understand the structure of the protein, to understand the dynamics of the protein. So as I said, all these structures will determine their function. So for a long time, there is something called structure function relationship. That means a function of a protein is determined by the structure of the protein. So in order to understand the function of a protein, you need to first determine the structure. And how do you determine the structure? NMR, XRD, cryo-EM, all this. So if you in biophysical chemistry, a large number of people solve the structures of the proteins. Okay. Then enzyme mechanisms and catalysis. That is what we have looked at, right? So you want to understand the mechanism of the enzyme. You want to understand how it catalyzes it. You want to look at the kinetics of it. So all this can be studied by this enzyme mechanism and catalysis. Protein folding, misfolding, aggregation. I'll spend more time on this. Okay. Folding, misfolding, aggregation. Then you have drug binding and inhibition, as I told you. So you have problems where you want to understand how a drug is binding. So one is enzyme mechanism, the other is enzyme inhibition, right? So you want to understand the drug binding and the inhibition of an enzyme by this. And then there are enzymes called molecular motors. So just like your, so if you want to take transport a cargo, right? So in your city, suppose You want to move something from one place to other. You have like a cargo transport, right? You have packers and movers kind of thing, movers. Like that in the cell also. So you think in the cell everything occurs by diffusion. So if a molecule has to be transported from one part of the cell is also pretty big, right? And compared to the size of the molecule, cell is pretty big. So if a molecule has to be transported from one part of the cell to other part of the cell, how do you think it will happen? It doesn't happen by simple diffusion. If a simple diffusion, it will take a very long time. It will be very random, right? You want to go from one point A to other point A. If you leave it to diffusion, diffusion will take it to anywhere. You all know what is diffusion, right? So diffusion is guided by random fluctuations unless there is a gradient. There are enzymes called molecular motors. Okay, just like your bus or just like your transport vehicles, there are enzymes that take cargo. That means cargo means certain molecules from one part of the cell to other part of the cell. And for them, for normal vehicles, what is the petrol? You have a petrol as the fuel, right? For these enzymes, ATP is the fuel, ATP or GTP. They hydrolyze, these are called mechanoenzymes. That means they convert the chemical energy from ATP hydrolysis into, so for example, you have a fan. Fan does what? It converts electrical energy into mechanical energy. Similarly, you have enzymes which convert chemical energy into mechanical energy, right? These are all called mechanoenzymes which transport molecules from one part to other. So these are different kinds of problems that people try to understand in biophysical chemistry. You want to understand how fast a motor is moving, right? For example, in our lab, we try to understand, we have enzyme called ClipX or ClipA, which kind of unfolds the protein and moves the protein from one part to other. So how fast this enzyme is moving. That means just like you have a speed for your vehicle, the enzyme will also have a specific speed. So their speeds are generally 3 to 4 nanometers per second. Okay, not kilometers per second per hour or something. The general speeds are 3 to 4 nanometers per second. Okay, so it uses, so you calculate the speeds at which these enzymes are moving. You try to understand all these things, and I'll spend little more time on these two, okay, folding, misfolding and aggregation. So these are various problems that people look at in biophysical chemistry or molecular biophysics. You try to look at these are the areas, these are the problems, and these are the major techniques that people use, okay. And this, as I said, this was generated simply using ChatGPT, but in general you can have multiple things. And you will be surprised to know, do you know a lot of Nobel Prizes have been awarded to biophysical chemistry? Do you know who got, what was Nobel Prize awarded to this year, not this year, last year, 2024 chemistry? Click chemistry, that was 2 years back. Last year, 2024 Nobel Prize in chemistry, who got, why they got? Quantum dots was 2023. You didn't give talk in your colleges, Nobel Prize talks? So last year the people got prize, Nobel Prize, so there are 3 people, basically Hassabis, Jumper and David Baker. So they got for computationally determining, basically using a software called AlphaFold and protein design. So what they did is, now you have, as I said, for determining the structure of the protein, you need to do X-ray, you need to do cryo-EM, you need to do NMR, you give a sequence and it determines it, right? But they have developed computational methods where you can just give the sequence of the protein, that means amino acid sequence of the protein, and that will predict what will be the structure of the protein. So till now you had to determine the structure by using cryo-EM, NMR, XRD, X-ray diffraction and all the crystallography and other things. Now these people, David Baker and Jumper and Hassabis, they have developed AI tools, artificial intelligence tools, and also protein design tools to determine the once you give the structure of the sequence of the protein, how it folds. Okay, so this was done. This was given Nobel Prize last year. Okay, and it happened very fast. So this entire discovery started probably in late 2000s, like even after 2010 or something. This guy is probably like very young, one of the youngest to get a Nobel Prize. Okay, so so that shows you how important it is. So now you don't have to actually determine the structure by using all the tools. By using computational methods you can determine. So this is what the Nobel Prize. If you look at overall, there are a lot of Nobel Prizes in chemistry that have been given to something related to biophysical chemistry. That means, for example, 2020, it was given to CRISPR-Cas9. So they are trying to look at basically protein DNA interactions. So they try to look at protein DNA interactions and they try to understand it, so that it was given. Then Henderson, Frank, they were given for development of cryo-EM. Cryo-EM, as I said, is used to determine the structure of the molecules. So cryo-EM of biological molecules, that is what it was given in chemistry. Then Martin Karplus, Malcolm Levitt and Ari Warshall, they were given for determining molecular dynamic simulations. So you can see, so for example, as I said, I don't know if you have heard of this guy, Ramakrishnan. So he's in UK, London, 2009. He got a Nobel Prize in Chemistry for determining the structure of ribosome, which is again a protein. So as I said, biophysical chemistry also deals with the structures of the proteins, right? So, and none of these, they are working on biological molecules, but most of them are chemists, okay? Most of them have a basic background. They are working on biological systems, but their background is mostly chemistry, or some of them will have physics, okay? So they got it in for determining the structure. So you can say quite a lot of chemistry prizes have been awarded to, for example, I guess you all know this, Watson Crick, Wilkins. So they were given, but they were given in physiology and medicine. That was for determining the structure of DNA. But in the same year, there was a protein structure, hemoglobin and myoglobin structure that was determined. It was given to Max Perut and Perutz and Kendrew in chemistry. So you can say a lot of basically chemistry prizes go to determining the structure-function relationship or dynamics, or as I said, dynamics means folding, or determining the structure, or determines, for example, this is structure, this is dynamics, right? This is protein DNA interactions, molecular interactions. So all these went to Nobel. So it's an area that is there for some time, but traditionally you will not see it in regular things, because a lot of people think that, oh, it is biology, but none of these people are actually biologists, or very few of them are biologists. Most of them are from a chemistry background, because chemists try to understand everything at a molecular level. They try to understand the interaction, they try to understand molecular interaction. So that is where a large number of Nobel Prizes have been awarded. Now I just want to take next few minutes to determine, so these are, as I said, these are some of the problems, trying to understand protein ligand interactions, trying to understand structure and dynamics of the proteins, trying to understand structure and dynamics of enzymes, protein-protein interaction, protein aggregation. Okay. Now, so one of the key problem that was there in biophysical chemistry is trying to understand protein folding. Okay, that means how a protein folds. So if you take a simple polymer, right? So if you unfold a polymer and leave it, will it always go back to the same structure? No, right? But that doesn't happen with proteins. Proteins, because the structure will determine the function, no matter how you unfold and leave it, the protein always goes back to the same structure, because the function is determined in the structure. This has puzzled the scientists for a long time. How can, like if you take any other polymers, every polymer will have a different structure, and once you, for a simple polymer you take and then leave it, and it will always go back to different structure depending on the thermal fluctuations and other kind of things. But for a large, people have observed that, actually it was first observed by Christian Anfinsen. I'll skip this. This is all you know. Okay, so this was basically observed by Christian Anfinsen, who got Nobel Prize in 1972, where he observed that once you unfold a protein by using various methods and then leave it, it always tries to go back to a same structure. Okay, and that means the information of the protein folding is in its amino acid sequence, right? Otherwise, so the folding process is being guided by amino acid sequence. Now, if the folding process is being guided by amino acid sequence, then if you just do a simple mathematical calculation, suppose let us take a very small protein. A small protein is 100 amino acids. Okay, generally proteins are very, very large. So a small protein is around 100 amino acids. Okay, 100 amino acids will have so there are 3 different conformations that are possible. So do you know what is how a peptide bond looks like? C double bond O, NH, then there is C alpha, then you have C double bond NH, right? So one angle is fixed. This C double bond O N angle, the peptide angle, that is always fixed because that is in a resonance, right? C plus O minus and NH. There is always a resonance there. So that is always in a planar geometry. But remaining all angles can change. So there are three different angles that are possible. So when the structure of the protein changes, all the three angles can change. So these are called dihedral angles or bond angles. So if you look at a simple amino acid, if you have n amino acids, you can have basically so each amino acid can change by around 3 possible conformations. So how much time does a bond rotation take place? How much time does each bond rotation takes? Bond rotation is guided by your rotational motion, right? You know that this is vibrational motion, this is rotational motion, right? Rotational motion generally of a bond, how much time does it take? So are you aware of various time scales? Vibration time scale, rotational time scales. You have done some spectroscopy? Okay, so if you are not aware, that's okay. So a bond rotation generally takes around picoseconds. You know what is picosecond? 10 power minus 12. So if the bond rotation can take around 3 picosecond, because there are 3 bonds associated with each peptide. So if you convert that, so number of conformations that are possible is 3 power 150, which is 10 to the power of 68. So if the protein has to fold by looking at all the possible conformations and trying to find the lowest minimum energy, then it should take such a large time, right? But actually the protein folds in within from milliseconds to seconds or minutes. So that means the entire information of a folding of a protein is inherent in its amino acid sequence. So this is a problem called protein folding problem, which has puzzled people for a long time. That means the entire folding process. So if each bond has to take a certain time, and there are 3 bonds that can change per amino acid. If you have a simple 150, I have taken 150 amino acid. So the number of conformations possible is 3 power 150. You know permutations and combinations, right? So if there are 150 amino acids and each amino acid can have 3 different conformations, what will be the total number of conformation possible? 3 power 150. So you have 3 power 150 conformations, and if each conformation takes around a picosecond, it would take around 10 power 48 years. So, but actually a protein folding process happens in about 0.1 to 1000 seconds. So it is very fast process. So, so the reason for that is a protein information is actually, so there is amino acid sequence that is actually guiding the protein through a funnel. So there are only certain restrictive dihedral angles or restrictive angles that will guide this particular protein into a certain native state, which is a minimal energy state. And you will be surprised to know that, so every protein cannot take, or every amino acids cannot take all the possible dihedral angles. Only certain dihedral angles are possible because of steric repulsions. And do you know who is the person who has devised a map which showed if certain dihedral angle, like for example, alpha helical structures will have a certain angle, beta helical structures, beta sheet structures will have a certain angle. Only certain, these are dihedral angles, okay. So only a certain angles are allowed is what this person has derived. Does anybody recognize him? It is written there. G.N. Ramchandran. Do you know him? G.N. Ramchandran did all this work right across here in Madras University. Okay. Very close to IIT. So G.N. Ramchandran was a faculty at Madras University who later moved to IISc. But all his work, he didn't get a Nobel Prize, but he did a lot of contribution which is worth Nobel Prize. If you talk to any biophysical chemist or anybody working on biophysics, people will know one person, that is G.N. Ramchandran. So G.N. Ramchandran determined what are the possible angles. So as I said, I told you this, right? So you have a peptide bond. This angle is fixed. C double bond to NH, this angle is fixed. This is a planar because this is in a resonance. If something has to be in resonance, that has to be a planar geometry. So this is fixed, but this angle can change, this angle can change, and so basically you will have phi, psi and theta, all these 3 angles can change. So which amino acids will change in which angle? what are the only allowed dihedral and angles, and if suppose something takes a specific angle, what secondary structure it takes, for example antiparallel, parallel or right-handed helix. So all this was devised and this was all calculated by this person called G.N. Ramchandran. In addition to that, he has determined the structure of collagen, okay, which is a protein, right? One of the first person to solve the structure of protein. So as such, biophysics or biophysical chemistry has a very, very strong connection to this city, because most of this top work that happened here in Madras University, which then he went on to do went to IISc. So at least you guys should know that a lot of good work in biophysical chemistry originated here. Okay, so if you next time when you see this person, you see his photo, just remember that he is a father of, you can always say that he is a legend in the field of biophysical chemistry or biophysics. Okay. Okay, so now I just wanted to, as I said, it is not possible for me to give information about a lot of questions, but I just want to tell you how important, how intriguing this problem of protein folding is. Now there is one more problem called protein aggregation. Now once you have a protein, right, if it folds correctly, then it will do its function, right, because protein structure is determined by the protein folding, and if it folds correctly, then it will do the function. But sometimes, a lot of times the protein misfolds, right. So if protein folding goes wrong, and that will give you what is called misfolded structure. So if a protein goes this, then it will give you what is called a misfolded structure, and once a protein gets misfolded, a lot of times this aggregates form. I'll give you a best example of protein aggregate, right. Now if you all eat egg, right. So egg has majorly, do you know egg has a lot of protein? Which part of egg has protein? The outer one, right? The liquidy part. If I'm talking about the raw egg, right? You heat it, it becomes what? It becomes a very white thing. That is nothing but it is a protein aggregate. So you are basically, there is a protein which is basically that kind of heating, the protein unfolds, and once it unfolds, basically it forms some misfolded structure, and then it all comes together and forms this white color thing which is called protein aggregate. So every day what you eat as a white egg, right? So it is not a folded protein, but it is actually a aggregate. Once it misfolds, and then it forms an aggregate of it. But that is fine for the eggs. But actually if it happens in the cell, if it happens in the body, it creates a lot of problems. Have you heard of diseases like have you heard of diseases like Alzheimer's? So what happens in Alzheimer's is, so there is a peptide called A beta 42, right? So this the function of it is not clearly known, but once this peptide is misfolded, it starts forming these large aggregates and it forms plaques in your body, okay, in the brain. So in the brain they will form these large aggregates called plaques. So these are proteins, so that proteins has to, if they are folding correctly, then they will give a function, but if they are misfolding, then they will start aggregating and they will form aggregates. A lot of aggregates. For example, you have heard of this disease called cataract? You haven't heard of cataract? So a lot of old people in their 65 or so, they develop cataract in the eye, right? I don't know. So the white color layer forms on the top of the eye, right? That is nothing but there is a protein called crystalline. So whenever a protein misfolds, there are enough enzymes that kind of folds. But once it starts misfolding, then it will start aggregating, right? So that white, the layer that you call, the people that ophthalmologists remove it, right, during cataract operation, if you see, that is nothing but a protein aggregate. It is an aggregate of gamma crystalline protein or a crystalline protein, not exactly. It is an aggregate of crystalline protein. So as long as proteins are functioning, they are very good. Your body functions very well. That it gives you a function, the structure. But once it starts misfolding, that means they do not fold correctly, then what happens? They will start aggregating. So once they start aggregating, they will create a lot of diseases. If you take a lot of neurodegenerative diseases like Alzheimer's, Alzheimer's is because you have a peptide called A beta that aggregates. There is a Huntington's disease or a Parkinson's disease. Have you heard of Parkinson's disease? And then cataract. All these diseases are because of protein aggregation. Now a lot of biophysical chemists also try to understand how is this, so can we reverse this process of aggregation, right? Can we reverse this process of aggregation and make these proteins again soluble? What is the kinetic, because at the end of the day these are nothing but your molecular interactions, right? Molecular interactions between the proteins. At a molecular level these are interactions between the proteins. So how can we weaken these interactions so that they don't form these large aggregates and they divide and form soluble proteins, right? Or how do we basically change these interactions? What are the kinetics of formation of this? How do we accelerate something or how do we decelerate something, right? You can also slow down this process of aggregation, right? So how do we decelerate something or accelerate something? So all this can be studied using your techniques of biophysical chemistry. In the sense you can, so basically all these physical parameters can be studied using these methods. Okay, for example, this one, this is the example. Protein aggregates forms a lot of these functions. And the last thing that I want to tell you in next just less than 5 minutes is, it is also important to understand, as I said, in the drug binding case, right? So you all have look. So most of the interactions that you study are basically from the energetics of it or molecular interaction. For example, you must have, you all know when COVID happened, there were no new drugs, but a lot of people tried to determine the drugs. How did they determine the drugs? So first they did was, so you have this protein ACE2 which is there on the cell surface, our cell surface, and this is the spike protein. You must have heard of this word spike protein. Spike protein is coming from the virus, right? This spike protein binds to ACE2 and it kind of interacts with that, and then the virus takes into entry. So this is nothing but a bunch of non-covalent interactions. Now when you are trying to develop a drug, what are you trying to do? You are trying to develop a molecule that will break these interactions. That means a molecule that can bind this particular receptor in a better way than the spike protein. That is how you can break the interactions between the spike protein and the ACE2. So in your drug discovery, the first thing somebody tries to do is, when you have, especially in terms of protein-protein interactions, you try to develop a drug molecule or you try to develop a molecule that change the energetics of the interaction and change basically the binding efficiencies of this. So in drug discovery also you will have a lot of studies on protein-protein interaction. So all these, as I said, most of these things are studied by physical chemists, or some of them are physics background, where you try to understand the physical properties of biological macromolecules, especially proteins, nucleic acids, to some extent carbohydrates and lipids. So this overall field of biophysical chemistry is applying the principles of physical chemistry, trying to understand, use these techniques to understand these particular problems associated with these focus areas. Okay, so I just wanted to give you an overview of this field so that you understand that it is not related to biology, but the molecules are biological, but the interactions are still chemical interactions, right? The interactions are chemical interactions, but the molecules are biological molecules. So you need to use spectroscopy methods or other kind of physical chemistry techniques to understand the various problems that are associated with it. Okay, so with this I'll stop here. Okay.
SwayamPrabha, Digital India, Educated India.